Okay, so today we're gonna talk about chapter 20, which is about cell communities, tissues, stem cells, and obviously a little bit on cancer towards the end. So this particular one, it has three main sections. We're going to talk about the extracellular matrix and the connective tissue where all the cells are kind of huddled together to make whatever organ system, tissue systems they are making. And they work together in that system. They communicate with each other back and forth in order to make it run the way it should be run. And then we're going to talk about specifically epithelial sheets and cell junctions, different types of cell-cell contacts, cell matrix interactions, and all that good stuff. And then at the last section, we're going to be focusing on stem cells and tissue renewal, how stem cells are utilized to make new differentiated cells to do the job they need to do in that particular system that they are in at that time. Some of this stuff we did before as well, but a lot of it is going to be brand new. So obviously, anytime we look at multicellular organisms, we are a little bit more complex than a single cell while doing the same function. So we have different organizations of tissues. Each part has a very specific function that it is supposed to perform. And so that kind of expert tissue for that particular function allows us to have a lot more control over how things work and what things can be done by the body. So vertebrates specifically like us have nervous, muscle, epithelial, and connective tissues. Those are the four major type of tissues that you can find. And these tissues, each one of them are not going to be just a single type of cells. You have to remember that they are going to be made up of multiple cell types working together in, combined together, joined together in this extracellular matrix, and they're kind of glued in with it. To form the final form that you have of that particular organ or tissue system and to do the work they need to do. The extracellular matrix itself is secreted by the cells. There are specific cells that can secrete a lot of the matrix proteins, and then other proteins can be provided by the cells, various types of cells that are present within it as well. And they serve not only to act as glue between that the cells to act, to keep them together, provide that scaffolding, but they also are the place for direct cell-cell and cell matrix contact. So they're, all the adhesion proteins that are present there, they are going to be helping those cells communicate with each other and communicate with the environment. So looking at that, here is an example for one of these tissue systems. And here you can see here you have the lumen of the gut. Here's a cross section of it. You see those, circular openings with these epithelial cells, and then your matrix back in the background, and you'll have connective tissue in between. If you look at the longitudinal section, you're going to find the lumen of the gut, right? This is that top portion with this nice organized epithelium layer. That would be right here. Right underneath it, you're going to have your connective tissue. This is going to have a lot of those sticky proteins that are going to be produced a lot of times by these specialized cells called fibroblasts that are going to maintain this extracellular matrix space. Then underneath that are your muscle tissues. So you have the various types of muscle layers. You have the circular fibers, the longitudinal fibers, and then you have another layer of this connective tissue, the extracellular matrix again, with the epithelium layer towards the inner side as well, that's facing the inside. So you have various layers in between meshed together. So let's talk in the first part about this extracellular matrix and the connective tissues that maintain these layers and maintain that structure inside our tissues and organ systems. So when we look at plant cells, obviously they're going to have different types of extracellular matrix than what we will have as animal cells. Part of it is because they have a very tough, that hard, woody exterior in the form of the cell wall, right? Cell wall itself is a type of extracellular matrix that's secreted by the plant cells to provide this really strong control on the cell shape and, provide it with that structural support. This kind of material can be, depending on the type of proteins that are secreted within that cell wall, And the type of cells that are present there can be either thick and hard, which is what you would see in wood, versus thin and flexible, which is what you would see in thinner parts of the plant, like the stems and the leaves. What that extracellular matrix is made up of to give it strength is the cellulose. Cellulose are essentially, they act like microfibrils, right? So they are cellulose microfibrils. They're basically long polypeptide chain, or not polypeptide chains, but long polymers of cellulose molecules chained together and then compacted to make a fibril, right? Kind of like you can think about it similar to the muscle fibers that we see in our muscles. Same idea, that the more of these chains are combined together, the more of these polymers or polysaccharide chains are compacted together, because it is a type of sugar, the stronger that fibril will be. So that's what you see here are your cellulose molecules that are going to be linked together, and then they are going to be compacted to create these myofibrils or microfibrils. These microfibrils are then interwoven with other sugars as well as structural proteins within the extracellular matrix or the cell wall area to provide more structural support and give it a lot of strength in that area. So here you can see that particular area. You have your cellulose microfibril, and they are kind of, mismatched in different directions to give you that strong structure. In between, you also have this other protein called pectin. Again, this one in plants can give it a lot of structure, and it's very sticky, adhesive protein. It's gonna kind of hold them together and bind all the stuff together within it. And then there are other sugar molecules, this cross-linking polysaccharide chains, that are going to be joining these different microfibrils together across from each other. Combined together, it's going to create a very strong structure, right? So you have the middle lamella, the primary cell wall, and then underneath you will have your plasma membrane. All of this combined creates that very strong structure. So looking at this, the orientation of these microfibrils are very important in directing the way the plant will grow and how it can interact with the environment, what type of stimuli can interfere with it or work on it. So the plant cells will control how the cell wall is laid down as they grow towards the light, for example, or as they are going into a structural support to maintain a particular shape. So here are some examples that will control it. So it's the turgor pressure on that particular structure that is laying down more cell wall components. And they can become perpendicular or they can be elongated. So depending on how it is that they need to do. And the cells are going to be basically growing perpendicular to these microfibrils the way they lay down. So if they are laying down in the from bottom to top, you're going to have growth in the length of the plant. And if they are laying down along the side edge, they will be growing towards the side at whichever side those cell walls are getting laid down. The other thing that you have is obviously along with these sugar chains, you still need structural support from the typical cytoskeletal structures. And here you have microtubules that help to decide which way these microfibrils are going to be laid down and how they are going to be organized within the plant cell wall. And that in turn will again dictate how the plant will grow. So if you look at the plasma membrane, and again, looking at that structure, this is inside the cell, right underneath the plasma membranes, you have the structural proteins, you have the cytoskeletal proteins, you have your microtubules, you have the connector proteins, you have all the good stuff, right? And then you have these cellulose microfibrils that are being added to whatever is already there, to the pre-existing cell wall. They are going to be interacting with the microtubule underneath. And as they do that by growing through a complex, a particular protein complex called cellulose synthase, which is where they are being made and added to your cell wall structure. And so wherever these synthase molecules are present, the cellulose synthase molecules are present, they are going to be adding these new microfibrils. They make them and they add them. And so as they add them, that's where the direction in which they will grow. Not every single one of them is going to be interacting with a microtubule, so it will not be building these microfibrils in that time. On the other hand, the animal connective tissue is a lot easier in some ways, a lot simpler in some ways compared to the cell wall structure as far as what it does initially. So extracellular matrix of the connective tissue is going to be providing that adhesion surface that the cells, the attachment points that the cells are going to be attached to. It provides a scaffolding, a structure for where the cells should go. But it's not to the same extent as the way the cell wall helps the plant grow. Cells of nervous, epithelial, and muscular tissues can be joined together with very little ECM because they can carry the mechanical load themselves, but the connective tissue itself needs a lot of extracellular matrix in order to spread out that mechanical load so the cells are not affected by them. So what acts as your ECM here, again, thinking about similar idea, similar structure, but very different component, is the collagen fibrils. Collagen is a protein. So unlike your cellulose, which was sugar molecules, right? Here you have collagen fibrils. Again, they're going to be long protein chains that are going to then link together with other collagen molecules to create this structure called, the triple-stranded collagen molecule. This is essentially the quaternary structure of this protein in its active form. These combine together, many of them stacked together, just like the cellulose, create a collagen fibril, and this collagen fibril is very strong and provides tensile strength to that connective tissue. Again, they're bundled, just like the same idea. Here in this cross section, you can see these bundles present, right? So you can see how many collagen fibrils might be present within a single unit. It is a little bit of, it can vary a little in different parts, but still within a section, you will have some good number of microfibrils, collagen fibrils combining together to create these structures. And all this empty space is not empty. It has a lot of these attachment proteins present that are going to help the cells maintain their structure and to be kind of combined within that tissue system. So how are they made? They are made initially with the procollagen molecule has areas within it that are not going to be part of that final structure. It has, the procollagen molecule is a lot longer and a lot more, amino acids in length. And it has signals for secretion. So it gets taken up by the secretory vesicles, and it is secreted as that procollagen molecule into the extracellular space. Once there are special proteins that are proteases, essentially, that cleave those extra extensions of the procollagen molecule, leaving it with just the main active component, which self-assembles into a fibril that can be anywhere from 10 to 300 nanometer wide. So they can vary in the size of those fibrils at the end of the day. The cells will organize this collagen that they secrete according to the tissue type that you're looking at and where you are. These are secreted by fibroblasts by large, right? And they are deposited in a very organized fashion. So typically they are going to be oriented and then rearranged according to the needs of the cell. The fibroblast will move across the collagen and pull it into a very compact sheet. So if you make a cross section in that area, you should see bands of collagen interspersed with bands of other extracellular matrix. In the skin, they're arranged in a woven pattern where they're alternating orientations of layers So in some layers, they are going to be longitudinal, and the others are going to be perpendicular, which is what will give you this classic look. In other systems, they are going to be aligned in parallel bundles, so they will all be in the same orientation. So now, once they are there, they obviously by themselves are not going to be enough. They still need to interact with the cells in their system, and they still need to be helping with the communication. And that's done by cells using specific proteins called fibronectin that are going to attach to the collagen with the help of another protein called integrins. So again, there are multiple proteins that are going to form together into this complex that is going to then help you interact with the collagen fibers and keep your cells attached and contained within that system. So if you look at the fibronectin molecule, it has cell attachment sites where integrin binding, and then it has extracellular matrix binding sites towards the N terminal that is going to be by binding to the collagen fibrils. So in the fibronectin, the amino end is going to bind to the collagen fibril, and the carboxy end is going to bind to the integrin dimers on the cell surface, right? So within, this is your cell underneath it. The cell itself is going to be bound to the integrins. We talked about how they bind to adapter proteins that are then interconnected with the actin filaments, keeping the structure of that cell intact, keeping all the proteins intact, keeping all the cells intact, and so cell shape intact. So this binding is essentially between the actin filament, adapter proteins, binding to the integrin that then in turn binds to this protein called fibronectin in the extracellular matrix. It's a secreted protein. And then this protein is going to have binding both the cells as well as the collagen on the N terminal. And this is going to keep it very strong binding modality. So these integrins can switch to an active conformation when they're bound at either end. Otherwise, they're going to be switched off. So your inactive integrin is going to be separate, so you'll have your alpha and beta subunits joined together, but they are not going to be in an active form. They don't necessarily bind to the cytoskeletal adapter proteins and the actin. However, when that binding to extracellular matrix takes place, that's going to make a strong binding to cytoskeletal structure where those actin and the adapter proteins. Similarly, if you have strong binding to the cytoskeletal structure, it can help promote binding to extracellular matrix where fibronectin and collagen. So cells, when we trypsinize them, we remove all the stuff from it, right? We remove the fibronectin from it. We are pulling them apart, so the integrins are getting kind of cut apart. So they're not going to have strong binding to the outside matrix, and that's going to prevent that binding to the inside actin as well. That's why they round up. They don't keep their normal, nice, clean, flat shape. But when you have that binding, when you have the attachment proteins there, and it starts to bind to that surface, it automatically helps to promote binding to the cytoskeletal part of it as well, making that structure strong and maintaining its shape. So next, we're going to talk about all the other sugar molecules and proteins that fill that space and resist compression, help to keep that cell from getting squished and getting destroyed, in addition to these integrins and the extracellular matrix protein like your collagen. So there are specific proteins that are called glycosaminoglycans, and they are exactly what they say. They fill up majority of that space, that what looks like empty space in the extracellular matrix and the connective tissue. Glycosaminoglycans are very complex, negatively charged polysaccharides. So they're just sugar molecules, sugar chains. They associate with proteins that can bind to sugar molecules, so proteoglycans, within the extracellular matrix, and they create with that binding a super large macromolecule. So when we have those cell culture treated plates, that's usually what they're getting coated with, is some glycosaminoglycans or similar stuff that are highly negatively charged molecules to help promote those bindings to happen. So here's an example of one of those, a part of that structure, what you might see, where you have these glucouronic acid and acetylglucosamine, and this disaccharide is repeated multiple times in a long chain in that structure. And so you have the carboxylant here, and they're all negatively charged. So the gels of proteoglycans, and glucosaminoglycans can work together to help to create this kind of squishy gel-like bed that is going to resist compression, and it's going to prevent the cells from getting too compacted. And this is an example of what that structure might look like. So here you have one of these proteins. So there's a chondroitin sulfate and keratin that can aggregate together and then join to the hyaluronan molecule, creating these aggregates. So you have your core proteins and then the linker proteins coming together to create this kind of caterpillar-like structure. And that structure is going to provide, again, a lot of strength to that system and promote resist compression in that system. So just a little review of this part. Extracellular matrix and connective tissues obviously are really important in maintaining the shape of tissues and organ systems, as well as organisms as a whole. Plant cells, you have that external cell wall made with cellulose microfibrils that give the plant cell wall its tensile strength. Similarly, in animal connective tissues, you are going to have the collagen fibrils that are going to provide the tensile strength. These collagen fibers, as well as the cellulose microfibrils, are organized very particularly depending on the position of the plant or position of the cell and how they're going to grow and develop. In plants, in animal cells, the integrins couple with the matrix outside to give that, they bind to the matrix outside and through the collagen, and to the cytoskeleton inside through the actin adapter proteins, to create a very strong binding to help maintain that shape of that cell. And finally, all the extra space within that connective tissue that is left behind after adding your cellulose microfibrils and the fibroblast cells is filled up with gels of polysaccharides and proteins that fill that space and resist compression. Any questions before we go to the second part? No questions? I feel like I'm going a little fast today, so I want to make sure you guys are with me. Okay. Okay, so just because you have the same biology behind that extracellular matrix doesn't mean that all cells are going to compact and come together the same way. They can pack together in very different ways to form different types of epithelial sheets, depending on the shape of the cell, the type of the matrix underneath, and the organization of those collagen microfibrils. So you can have columnar cells or squamous cells, right here, where you have these cells directly attached to the basal lamina in this way, or you can have stratified and cuboidal cells here attached to the basal lamina. Do you see how very different they look? Columnar cells will many times have an apical and a basal side that is very different in the type of proteins that are present in the membrane and in their function and permeability. And that helps them perform some cool functions because of that. So epithelial sheets are polarized and they rest on that basal lamina, which is again going to be connected to the connective tissue. Epithelial sheets will have, like I said, an apical and a basal side, and the more length you have, the more distance you have between those sides, the more different they will be. So you have up here is your free surface with the apical side, and down here is the basal side that's connected to the basal lamina, which in turn is connected to your connective tissue. The basal lamina is going to be what provides support to the epithelial sheet and keeps it in that structure, maintains that structure. It's very important for that. And then it leaves that apical side, available to react to the environmental stimuli, to whatever is present in that environment at that time. Within there are tight junctions that allow sheets to serve as barriers between different types of solute diffusion. So the tight junctions are going to create basically zippers type things, right? So they are seals that will prevent different parts of this membrane from cross-talking between each other. So right here, these are two adjacent cells, right? And these are the membranes of the two adjacent cells. Right here, you can look at it laying down, laying flat. You have a tight junction up here. You have a tight junction right there. In here as well, there's a tight junction right here. In that top surface above the tight junction, you're going to have different biology, different permeability, different proteins present than what you will have in the bottom side, in the opposite side. So each time there's a tight junction, it can create a very different environment in between those tight junctions. And within those areas, within each area that is open and interacting, you will have those plasma membranes interacting with each other, cross-talking with each other, and creating a homogeneous environment. So here you see these tight junctions kind of sealing off different portions. These tight junctions will go all along. It's not just like at the surface, it's going to be all along that 3D cell membrane. And so it's going to seal off a portion. It's kind of like taking those, the heating things to seal your little Ziploc bags in various parts to create compartments. That's what you're doing here. So these junctions are made partly, the seals are made by a protein, a couple of proteins called occludin and claudin. So occlude, right, to seal off in this case. And these are going to be rows of these proteins that are going to create a very tight seal. So looking at that a little bit more closely, you can see that they are going to have that tight seal. So other types of, sometimes you don't need a super tight seal, you just need them to interact with each other. So you can instead have cytoskeleton-linked junctions that are going to bind at just simply cells that are adjacent to one another. There are two different ones that can happen. One is adherence junctions and the other ones are desmosomes. Both of these use the same linker protein called cadherin. In adherent junctions, the cadherin molecules outside the cell are tethered to the cytoskeletal actin filaments inside the cells. While in desmosome, the cadherins is not bound to actin, but rather the intermediate filaments inside the cell. So depending on what type of cytoskeletal structure they are going to be interacting with in the inside, you're going to have different types of sealing and different types of interactions. So adherents are going to be with actin and desmosomes are going to be with intermediate filaments. Here is an example of how this will look differently in, how they behave differently once made or built properly. So here, again, these are some columnar cells. These will have a clear basal and an apical side, which is facing the lumen of the, let's say your GI tract. You have all these microvilli at the apical side that are going to be taking in all the nutrients and all the glucose and everything from the environment. These microvilli will have that structure maintained and have the movement using actin filaments that are going to be in those inside the microvilli, and they are going to be helping with that structural integrity and movement. These actin filaments are going to be interacting with other bundles of actin filaments within the cell towards the area where the adherent junctions are. So the adherent junctions, here they are. These are all your proteins, right? These are your cadherin proteins. They are bound in the inside to actin filaments that are creating a band across the membrane, across the epithelial sheet at that point. So you have tight junctions here, which are going to prevent the proteins from up here to go down here, creating that disparate biology within the membrane. You have these bands of actin filaments that are joining the entire structure together, creating it as a unified epithelial sheet. And these are bound by the cadherins within those adherent junctions. So they together will form this adhesion belt all along that sheet, keeping it together in that form and keeping that top half separated from the bottom half, from the basal side, where the cells are not interacting with each other. All the interactions are on the top portion. This type of epithelial sheets, once made, are going to be pretty strong. You can imagine it's like, it's like sewing them, stitching them together to make a very nice, clean structure. They can be bent to form a tube or a vesicle. And here is an example of that, where you can have invagination of these epithelial sheets to create epithelial tube, for example, or another structure. Neural structures are formed with something similar to that, right? When epithelial sheets come together to create these kind of vesicles during development. So they will have a similar structure. On the other hand, desmosomes work differently. Okay, so in desmosomes, you are going to link keratin filaments of one cell to the other using the adherent proteins as your, again, the binding partners. So in this case, you have the adherent proteins at the cell surface, and they're all kind of clustered together in the cytoplasmic plaque that are made of intracellular linker proteins. These linker proteins, this kind of plaque-like structure is in turn bound to many keratin filaments. They're anchored to it. So that's how they're going to be connected. It's not a direct binding between the actin and adherent, or in this case, the keratin and the adherent, but it is through the presence of those intracellular linker proteins making that cytoplasmic plaque. So again, this is going to create a very strong structure in here, very strong binding between these two cells, adjacent cells, allowing them to have some kind of crosstalk and communication between them. But this one is going to be all across that cell, right? So the entire cell has those in a very strong way. Another type of desmosome, this is a subtype of desmosome, is what we call a hemidesmosomes, which anchor these keratin filaments to the basal lamina to keep that basal side away from the apical and to keep it structured to that, tethered to the basal plasma membrane. And in this case, this hemidesmosome, the keratin filaments are still attached to the plaque of linker proteins, but the linker proteins are not attached to adherent, but rather to integrin proteins that are integrated into the basal lamina. Finally, the fourth and last type of big types of junctions that interactions that we're going to talk about are gap junctions. These provide neighboring cells with a direct channel of communications. So all the others, they were binding with attachment proteins. There wasn't any interaction. There wasn't any movement of molecules from one side to the other, right? If anything, they were stopping that movement of molecules from one place to another. So there was no crosstalk between the cytoplasm in those cases. However, in this case, the gap junctions provide little, essentially, cores that adjacent cells could have interaction with each other through the plasma membrane. So it's a direct channel of communication, direct line of communication between interacting cells, between two adjacent cells. So here is an example of a gap junction. These cells are going to be joined together with these, connections. They're essentially called connections. They are composed of 6 protein subunits coming together to create this 1.5 nanometer pore-like structure or channel that can then help to exchange cytoplasmic fluid and thus transfer information from one place to another. These are essential in movement of signals that are from extracellular systems into the cell because those are regulated by permeability of your gap junctions. So here is an example of that. So if you give, so here is an injected neuron, you're looking at neurons labeled through the gap junctions. Before dopamine, after dopamine, which would cause this to be disrupted, you have much less movement within that structure because these gap junctions are no longer functioning, and so that crosstalk is unable to be executed. So at the end of the day, here are your different types of structures that you should know about, and animal cells that are going to help to join and communicate between adjacent cells. You have tight junctions that are going to tightly seal neighboring cells together in the epithelial sheet and prevent leakage of any extracellular molecules between them. They help to polarize the cells and keep the permeability intact, difference of the apical versus basal lamina intact. Then you have adherent junctions that are going to join with the actin bundles and create a strong band to provide strength to that epithelial sheet, keeping it together. Then you have desmosomes that are going to be bound with the intermediate filaments in one cell to those in the neighbor, and they are going to be a little bit more loosely attached in this case. And then you have gap junctions that are going to form these small channels that are going to allow for communication between adjacent cells in the form of cytoplasmic exchange in organic molecules, cytosome, different signals, anything you can think about. And finally, you have those hemidesmosomes that help to bind the cells to the basal lamina through binding with integrins within it. Questions? No questions? Are the junctions in that order? So, the cells will typically have something very similar to it, especially if it is a polarized cell. If it is not a polarized cell, no, they do not have to be in this order. They could be in a different order. But typically you will, in polarized cells, like the ones that we see in those epithelial sheets within our GI tract, within the neuronal system, that's the way that it's going to be, yes. So in plant cells, you also have some additional connections. Obviously, plant cells cannot have these junctions like we have all these different types of interactions like we have in animal cells, because they have that really hard outer surface, right? They have those outer structures in there. They have their cell wall, and that is not going to be permeable to these tight junctions and adherents and all that good stuff. So in there, they have an alternate form of cell-cell communication and cell-cell contact system, and that is by plasmodesmata, or through plasmodesmatas rather. Plasmodesmatas are functional counterparts of essentially gap junctions. So they work the same way, but through the plasma membrane, or not plasma membrane, cell wall rather. I apologize. So here is an example. You have these cells in here with their cell walls, and within the cell walls, they have several areas of break, what looks like break. If you look at it carefully, and if you look at it more in detail at a macro level or a micro level, you will see that that is an area where the smooth endoplasmic reticulum from one cell is interacting, is kind of pushing through on both sides and creating a channel of sorts. This structure is called a desmotubule. These desmotubules, again, provide a channel of communication between adjacent cells and keep them interacting with each other and creating that surface where they can have that crosstalk. There are many, you'll find multiple plasmodesmatas between adjacent cells so that they can have several areas of communication. So epithelial sheets are polarized and they rest on basal lamina. Tight junctions allow that separation to be present so that they can have that polarization. They create a leak-proof separation between the apical and the basolateral cell surfaces. The cytoskeleton-linked junctions, like your desmosomes, are going to allow for, and adherent junctions are going to allow for cell-cell contact and keeping that structure together. Gap junctions, on the other hand, are going to allow for cell-cell communication with the transfer of cytosolic inorganic molecules, small molecules, signals, cytoplasm, etc. Okay, gonna pause for a second here. So no questions so far? We're good? We're doing good on time, surprisingly, today, which is great. Okay. Okay. So in the last part of this chapter, they talked about stem cells and tissue renewal, how those systems are continued and how those systems are maintained throughout our lifetime. So at the end of the day, like if you think about it, that one single fertilized cell is going to create, that one single cell is going to eventually create that entire organism full of all the different types of cells that you see, right? And so that genome that is present inside that fertilized egg is going to eventually determine what structure you're going to see. So looking at these two fertilized eggs, they look very similar, but eventually are leading to very, very different organisms at the end of the day once they're fully developed. In these structures, as they're developing, you are going to see a mixture of many, many cell types. And we already kind of talked about it a little bit in the last section, where we are thinking about tissues, and tissues are not just, so if you're looking at a piece of liver, it's not just going to be liver cells, right? It's going to have those hepatocytes, the main liver cells, but then it's going to have your, extracellular matrix and the fibroblast. It's going to have the connective, the blood vessels going through it to provide it nutrition and oxygen. So it's going to have those epithelial layers, the blood, different types of blood cells. Then you're going to have the nerve cells that are communicating signals to that tissue. So it's going to have all the cells associated with the nervous system. Then it's going to have Kupffer cells or other helper supporter cells in there. There might be some lymphatic vessel coming through it to help to keep the balance, right? So it's going to have many, many, many different types of cells within that tissue system. It's not just going to be one single type. And all of them have to be able to communicate with each other in order to work in unison and do the work they need to do. So typical tissues will have an epithelium or epidermis layer. They will have some loose connective tissues that's going to have a lot of typically blood vessels and sensory nerves to communicate with the outer environment. And then on the other end, you through the dense connective tissue of the dermis and towards the fatty connective tissue at the bottom, you will have the base nerves and blood vessels providing it with the nutrition that it needs. So this is just looking again at the cells in the skin in this case. So you have many, many layers of skin cells. And you would see that, on the top, you obviously have your dead cell layers. But even once you get to the actual keratinocytes, you're going to see multiple layers of live keratinocytes before you get to the area where you have actively growing, dividing cells. Those are going to contain not just your keratinocytes, which are your main skin cells, right? But it's also going to contain other types of cells, like your Langerhans, which are your immune system cells, and then melanocytes that are going to produce the pigment, giving you the skin color that you have, and all that stuff in between. Underneath, so that's all that pink layer. Then when you get underneath it to the connective tissue, this is that live connective tissue that's going to provide you with the support system for these cells to get attached to. This is going to, again, contain your fibroblasts that are going to secrete the extracellular matrix that the cells are going to be under attached to. You'll have the collagen fibers that are going to provide that tensile strength. You will have some immune cells typically with those lymphocytes and the macrophages, and then you'll have your capillaries providing the oxygen and the nutrition to your skin cells. And so those would be right here. Here's an endothelial cell forming capillary. And then further down, when you get to your dense connective tissue, that is going to be more of that strengthening component. So this is more living, this is more strengthening. And this will contain a lot more collagen fibers cross-linked in various directions to provide the tensile strength. And that have to be there to provide that extracellular matrix, secrete that matrix that these are all going to be joined to. So where do all these cells come from? Well, most of these cells are going to be coming from, well, all of these cells at some point are going to be coming from stem cells. Stem cells are different in different types of development, at different times of development, and even in different tissue systems. So every tissue system that we have, every organ system that we have has its own subpopulation of partially differentiated stem cells. Those are not totipotent cells. A totipotent cell is a cell that can be anything all the way up to a full organism, but rather there might be pluripotent cells, or even more specific than that. So a pluripotent cell can be a few different types of cells, but it can never give you a full organism. These little populations of stem cells in those tissues are going to be specific to the tissue itself, and they can undergo some limited cell division to renew themselves, renew their own population, so they keep a little bucket of themselves. And then as they receive signals from their surrounding tissue, from the environment, they can become differentiated. And as they become differentiated, they are going to divide, and they are going to create, they will divide with those differentiating signals, leading to terminally differentiated cells of the various types that are needed, whether it's hepatocytes or fibroblasts or something else. These terminally differentiated cells will no longer undergo cell division, will do the work they need to do until they die out, and then more cells will need to come in to replace them. So the stem cells are the ones that are renewing the population of this, whatever that organ system is that you're working with. They are getting differentiating into terminally differentiated cells that are going to develop from these precursor cells. It's a dead end for those cells. They're not going to continue dividing once they're terminally differentiated, and they will need to be replaced by more stem cells when you get to that road. So looking again at an epithelial sheet, here in this case, you're looking at epithelial lining of the intestine. So remember, you have those microvilli as well in there, and you have these sheets of cells in there. You will have, if you look at this particular structure, you have the crypt, which is where a lot of these stem cells are, right? Down here at the bottom of the crypt. And they are gonna, as they divide, they move up this crypt, this little structure. And as they divide, they are going to become more and more differentiated until they reach to this microvilli state, where they are going to be fully differentiated, non-dividing, terminally differentiated cells. And within that structure, they're going to be different types of cells. You could have the mucus-secreting goblet cells. You can have the absorptive brush border cells. You can have the typical villi cells, whatever they are. So these are the various types of cells that are already differentiated. And down here in the crypt, next to that connective tissue, is where you have the dividing stem cells that are developing these cells, right? So this process takes 3 to 6 days in humans to go from that bottom all the way to the final destination where they are going to be at, okay? And if we take a cross section of the various part, you will be able to see where the differentiation is happening and where the terminally differentiated cells are towards the top. In similar idea, the epithelial layer inside our epidermis, the skin, it grows the same way. It's renewed from stem cells that are present within the basal layer. And those basal layer cells, they are right above that basal lamina, right? So this is your basal layer of keratinocytes, and these are the ones that are going to be dividing. And once they divide, as they move up again, they are your epithelium, right? The epidermis layer. These are your living cells. They are getting older and more, and as they grow old, they start to become deflated. They start to lose their structure. They start to lose their organelles, and eventually you have those dead cells that are just flattened cells packed with keratin filaments and nothing else. That will have several layers of it before it is finished. So here, if you take a cross section of that, this is looking at an H&E. Right down here, the dermis is where you have active cell division happening, and that's right above your connective tissue. And this epidermis is living cells, but these are the cells that have already terminally differentiated. And on top is your dead cells, dead cell layer. In blood, you have a similar idea. And so in blood cells, right, in the bone marrow, You have your stem cells that are going to differentiate into various types of cells, and those are controlled. The percentage of what type of cells are going to be made at any given time are controlled by the signals received in the body, by the amount of oxygen you're taking up, by the amount of red blood cells that are present, whether you got exposed to the virus or the bacteria of the day, whether you have an infection or cancer, whatever the case may be. And so the same stem cells, depending on the need, can differentiate into any of these different types of cells, whether it's an erythrocyte, a platelet, or any of the immune cells like your basophils or eosinophils or neutrophils or lymphocytes. So here is an example of that. You have your hematopoietic cells. These are in your bone marrow. They can self-renew and make more stem cells, but once they receive the differentiation signal, they can give rise to any one of these many different types of cells related to the blood. So if you were to take an embryonic stem cell, which is a lot of what the a lot of stem cell research comes from, when we take that, we do not take cells that are totipotent, but rather we take them from an early embryo at the blastocyst stage, where you have a cells of inner mass in there that are that can be any of the tissue systems, but will never give rise to an entire organism by themselves. These cells from the blastocyst are cultured in dishes, just like what we did in the lab, but with very specific differentiation signals to create different types of stem cells, different types of cells, differentiated cells. These cells can be made into neurons or fat cells, macrophage, skin layers, cardiac tissue, any of those things. So these type of stem cell technology is what we called, stem cell research. Another type of stem cell research is done by taking embryonic stem cell-like structures. So we just take, in this case, cells that don't necessarily come from even the stem cells. You can actually take components of these stem cells from different organs as well. And they can be called induced pluripotent stem cells. So those cultured embryonic stem cells can be induced based on what you give them, what signals you give them, to make different structures, different tissue systems, different cells. You can also sometimes take specific types of normal cells and induce them to become pluripotent stem cells by de-differentiating them. So these induced pluripotent stem cells are a very convenient source of human embryonic stem cell-like cells, and they can be used to form organoids in culture and are essential for example, organ transplant research to try to find better ways, easier ways that do not require other human sourcing to create the different types of tissues and organs needed for survival of patients that don't have working organs themselves. So here you have those aggregates of cell culture cells that have been stimulated to create neural cells, and then as they develop, they become into a neural retina in this case. So they are making eye-like structures in this case. So stem cells are are very important in today's research for organ transplantation, as well as just understanding how different tissues and organs work. Tissues are organized by mixtures of many different cell types, not just one type itself. And these different tissues are renewed at different rates. So each one of them will have their own rate based on the cell signals received by that particular tissue system. Stem cells and proliferating precursor cells can generate a continuous supply of terminally differentiated cells, and the stem cells are the ones that can be regenerated themselves as well. Each organ system in our body maintains its own stem cell population to do this job. So it's not just like, that they no longer exist. They are always there. And they are the ones that are used by our bodies. And they do reduce in number as we age, but that's what's getting used by our body to differentiate and get more replace damaged organ systems. Induced pluripotent stem cells from the blastocysts provide a convenient source of human embryonic stem cell-like cells that can be used to form organoids in culture. Any questions?